So the first speaker is Aaron Debon from ETH Zurich, where he is finally a PhD student in Don Hilbert's lab. His main interests are finding new ways to probe biology and create new functions. And with that in mind, he's been exploring microfluidics since his bachelor's studies at ETH, and now he's moved to applying directed evolution in his PhD work. And today what he's going to tell us about is about how he has applied microfluidic droplet sorting strategies to engineer oxidase enzymes and other classes of enzymes. On a personal note, Aaron enjoys hiking, so I hope you had a really good weekend hiking in the mountains. So without further ado, Aaron, the Zoom is yours.
Okay, I'll start sharing my screen. I hope you can all see my presentation. Great. Well, thanks for the introduction, Anya, and thanks for organizing this seminar here. So as we've already heard, my talk's going to be about ultra high throughput screening with what you see in the background moving here. These are aqueous droplets in an oil phase. And what we have done is we've used these droplets to engineer oxidases. Oxidases and other enzymes, of course, are a prime target for green chemistry, and in the industrial chemical manufacturing, we can use them to make catalysis more environmentally friendly. And first, I'm going to show you 3 examples of compounds that people have used oxidases to make building blocks to make these compounds. The first one, boceprevir, which is an HCV inhibitor, protease inhibitor. We have this bicyclic amine here, which the Turner lab and Merck have used MAO-N, so monoamine oxidase from Aspergillus niger variant, to desymmetrize this molecule with very high specificity. And the second one, solifenacin, we have a deracemization, which is done by combining an oxidase, here the same as before, another variant though, with a reducing agent which is non-specific. So we can, by specifically oxidizing the racemic mixture and non-specifically reducing the product, the amine we can form, we can deracemize a racemic solution. And in the last example, which is a recent example, which most of you will probably be familiar with, is galactose oxidase being used in a cascade which almost synthesizes all of this molecule as islatravir by enzymatic steps. And the first step is galactose oxidase desymmetrizing this alcohol here. And with enzymes, we can carry out reactions at room temperature, aqueous solutions, and also make chemistry that is very difficult to carry out by normal synthetic methods. The only problem is, as I've already pointed out, none of these enzymes are wild-type enzymes. All of these enzymes had to be engineered and evolved towards being useful in the given conditions that these reactions take place. And I just wanted to give you the number of mutations that people have put into these enzymes to adapt into the process, and it's a fair amount of mutations that you have to put in. So we thought that by speeding up the way we screen and the way we evolve oxidases, we would provide a further method to enhance the usage of oxidases in industrial biocatalysis. And the way we end up with all of these mutations, all of you are probably going to be familiar with, is directed evolution, where you take a wild type or a variant, we select certain residues that we want to target for a mutation. And we have to make every single of these variants to test them in the end. And we have to gauge the fitness or how good our enzyme carries out the function that we're interested in. And with that process, by going in cycles, we can slowly climb the fitness peak that we want to climb. And our desired way of improving or making the cycle go faster is, of course, by increasing the throughput and by applying design strategies of libraries that fit our throughput. And you can see here how oxidases are usually being screened. We can have a microtiter plate assay where we isolate single clones into wells on the 96 well plate, as seen here. We have colony-based assays where we stain colonies that are partially lysed and we spray them with the substrate. And what we are going to add here is a microfluidic approach where we have a schematic droplet here, as you've seen on the title slide, which acts as a boundary for diffusion of the genotype and the phenotype. And this way we can, in principle, screen millions of enzyme variants in a single experiment. And what's special about microfluidics that oftentimes people use tagged substrates. So they would have a fluorescent tag on here, it would be cleaved, and we would measure this tag. But with oxidases, we have a kind of unique outset here that the oxidases recycle their cofactor using molecular oxygen and produce hydrogen peroxide in the process. And we can use a coupled enzyme assay to monitor this reaction through the side product. And this way we are uncoupled from the substrate. We don't need to tag the substrate. And also any oxidase will produce hydrogen peroxide if it carries out the reaction. So we can also screen any oxidase in principle. And the enzyme that we chose to evolve was cyclohexylamine oxidase, short CHOW. It does what the name says. It oxidizes cyclohexylamine, and it does so at around 10,000 per molar per second in kcat over km. We chose a substrate, which was the 1-phenyl tetrahydroisoquinoline. And what we wanted is an R-selective CHOW variant, which was not described in literature before. Actually, no activity towards this compound was described in literature before, even though it has been tested. And through having an R-selective variant, we could make the S enantiomer of tetrahydroisoquinoline variant here, which would be a building block of solifenacin, which I showed you in the first slide. And this is how we're going to sort, or how we're going to assay these droplets. We call it fluorescence activated droplet sorting, or short FADS. And we have microfluidic chips where we have built in certain functions that serve the purpose A of generating an emulsion, which you can see down here. This is happening at a speed of around 1 kilohertz. So this is slowed down a fair bit. You can see an oil phase coming from here, an aqueous phase coming from here. It's being sheared off into droplets. And what we can do is, here in the stream, we can combine multiple aqueous phases. In this case, we have our cells and we have our reactants. And the reactants contain the substrate and HRP and the detection cascade and lysis agents. So we're going to break open the cells in these droplets to release the enzyme. And if we dilute down the cells low enough, these droplets should contain single cells, so we can assay single variants. Then in the second function, we have to incubate the droplets for a certain amount of time for the reaction to take place. So you can see here. So you can also see that these droplets are very homogeneous, which is important. So we have very similar reaction conditions for each variant that we test. And at the very end of the chip, we have a sorting junction, which you can see here. And what we do is we measure the fluorescence with a laser at the point here. And if the fluorescence exceeds a certain threshold that we set, we will give an electric impulse to this electrode, which will deflect the droplet to the positive side. And these droplets will, of course, gather and analyze further. So we wanted to update the library design a bit for this given task, because we have certain features in our enzyme that we can harness to make library design more specific, the active site is completely internalized into the enzyme and very hydrophobic. So what we did was we designed new codons, DYT and BYT. They code for different size of aliphatic side chain residues and residues that can hydrogen bond. And these are the DYT codon are targeting all of these residues, whereas for this proline here, we decided to use BYT, which also codes for proline. In case this proline had a structurally important role, we could conserve it during the evolution. So what this yields is 8 residues that we target all simultaneously, and we have 1 million members in this library. And since every single one of these amino acids is encoded by just one single codon in these codons, we have no stop codons and we have no bias towards one residue. And now everything is coming together. We have the substrate that we chose. We made a library, and what we did is we encapsulated them on chip and we incubated them to have the reaction cascade taking place. And after sorting, we collect the positive droplets. And what we then can do is we can either iteratively sort to enrich the library more, which is what we did 3 times. And we can also analyze the clones, single clones after every round. I'll give you an example how this would look like here in the microfluidic sorting device. On the y-axis, we have a barcode dye that we add to all of the droplets so we can detect all of them. And on the x-axis, we see the activity, so the reporter dye. And we are interested, of course, in droplets that contain more reporter dye than the ones that probably contain no enzyme or are dead clones. And on the right-hand side, you can see here a plate assay on top of the unsorted library. And as you can guess, of course, it's very unlikely to find a very active clone out of 1 million clones. And here, after three rounds of enrichment, we can see we find many active clones. And what we did is we picked the most active clone off of this plate and we analyzed it. We sequenced it and we found that it had 5 mutations. So here you can see the wild type crystal structure, and here a computational model that we made by in silico mutating these 5 residues and then docking in the substrate. What you can see is that with reducing the size of these residues here, we've created a second pocket where this isoquinoline ring can fit in. And here we have 2 additional mutations on the exit channel. So the substrate is believed to come in here and the product is believed to leave at the same place. And this one is in the literature. The methionine to threonine is described to enhance substrate uptake. We can also see the positioning of the substrate with the hydride that's going to be transferred to the FAD being placed in good proximity towards the FAD and the active center here. We've kinetically analyzed this variant, and what you can see in red here is the wild type. You can see that for the R enantiomer of 1-phenyl tetrahydroisoquinoline, we have fairly low activity, and for the S enantiomer, we have no detectable activity at all. But after just 1 round of evolution, we've improved the kcat over km of this enzyme by almost 1,000-fold. And furthermore, the selectivity of PT1, the variant that we created, has an S factor of 4,200. So it is very specific for the R enantiomer, which is exactly what we desired. And as a quick comparison, again, we have the wild type with the native substrate cyclohexylamine, which has a kcat over km of roughly 10,000. So we are actually in the range of a wild type enzyme here within a single round of mutagenesis. We were interested in analyzing our model, how we could explain this huge selectivity, and what we found through docking the less desirable enantiomer is that compared to the R enantiomer here, we have a bidentate hydrogen bond being formed with the newly mutated serine residue here, and with the backbone here, which forces the substrate into a conformation where the hydrogen points away from the FAD, making it catalytically non-relevant. We used our PT1 variant then to carry out this deracemization reaction with phenyl tetrahydroisoquinoline. We challenged it with the racemic substrate, and we added the borohydrate here as a reductant. You can see the wild type, which we've even measured for multiple days afterwards. There's no enrichment of the S enantiomer in this reaction at all, whereas the PT1 variant carries out this reaction very selectively. It reaches 99% EE after approximately 9 hours, and this all happened at 0.05% catalyst loading, so it's also very efficient. And if we analyze further the substrate scope of these enzymes, you can see in red here the wild type, of course the activity for the wild type for the native substrate, and here for primary amines we can see for primary amines with small substituents here we have high activity for their S enantiomers, and as we grow this moiety here, for example N-propyl or this parachlorophenol, we lose a lot of activity. And what we've seen with PT1, we have completely changed the activity profile of this enzyme, and we've killed it for its native substrate. And we see that we also diminished the reactivity towards the small primary amines a lot. But as we then incorporate incorporate bigger moieties over here, we restore the activity. Also, we see that we have a switch in enantiomer selectivity. As you can see here, we prefer the R enantiomer over the S enantiomer, and the same here. For secondary amines, we can see that we have low activity for all of them with the wild type enzyme. However, through evolution, we've of course already discussed this part with the phenyl tetrahydroisoquinoline. We also have increased activity for 1-ethyl tetrahydroisoquinoline, but almost no increase in activity towards the methyl substituent here. So for some of these substrates, we only had racemic substrates available. So we were wondering what is the selectivity of these. What we carried out was further deracemization experiments where we combine all of these. Substrates with our enzyme and the deracemization compounds, and we analyzed with chiral HPLC after a certain amount of time what the enantiomer selectivity was. And we can see, as we've observed for this one, we have a selectivity switch for the R enantiomer, but it's fairly unselective. We only have 50% EE here, whereas for the methyl one, where we saw no increase in activity from the wild type to PT1, we have completely unselective reaction here. Probably the reaction is not really taking place. For the ethyl substituted tetrahydroisoquinoline, we restore activity and also selectivity and are able to recover this at high yield and high EE. And we cannot distinguish the chlorine substituted benzhydrolamine here. We cannot distinguish a chlorine hydrogen group here. So I already come to my conclusion. What you saw is we have this microfluidics as they developed. We screened around 1 million variants of cyclohexylamine oxidase with it, and we found this new variant which has 5 mutations. In a single round of evolution, we took an enzyme that was unreactive towards our substrate and made it into a viable catalyst that could be used for synthesizing 2 APIs in very high stereoselectivity. So with this assay in hand, we have future targets. So A, we can use it to further diversify cyclohexylamine oxidase because we already have a library in hand. And for example, this piperidine is one methyl substituted piperidine is completely inactive with the wild type enzyme, whereas we already detected activity by screening our library. And we want to see if we can distinguish the chlorine from the hydrogen here. It would also be interesting to go into the and screen tetrahydroquinolines with this assay. Also, we can use different oxidases. Glucose and galactose oxidases are well described in literature. And when you do a quick blast search of Chow, for example, you can see a lot of predicted oxidases which haven't been characterized yet. So this assay could also be useful for screening metagenomic libraries. So with this, I want to thank my advisor, Don Hilvert, for having me as a PhD student, my collaborators and people that continue this work, our whole group here in Zurich, and SNF for the funding. And then I'm open for any question. Of course, thanks for the invitation to present here.
All right, let's give Aaron a virtual round of applause. Thank you so much for a great talk. As we're waiting for questions, maybe I can ask the first one since I'm very interested in oxidation reactions. Is oxygen delivery and the cofactor delivery an issue for the microfluidic system when you evolve the enzymes?
Not that I know of. So in this case, we didn't add any more cofactor at all to the reaction. Yeah, it's just there from purification. Yeah. Of course, you could add more cofactor into. If you look at the design of our microfluidic chip, of course, you could imagine adding more cofactor to your reactant channel. Of course, cofactor uptake will take some time probably, but incubation can be multiple days even. So I don't know if that answers your question.
Yes, thank you. I guess we need to see how it's going to work with that other example. Thank you.
Okay, Aaron, we've got multiple questions coming in. So what I'm going to do is I'm just going to allow people to speak. So first of all, I have Peter Wanyama. You should now be able to talk.
Are you able to hear me?
Yes, we can hear you now.
Okay, yeah, the first question, just out of curiosity, how did you determine the stoichiometry of your enangiopure compounds? Then two, when you identified the gene that were responsible for that, is there a way you can play with the system so that you get the opposite stoichiometry?
Okay, yeah. So the first question, for these two substrates, we simply bought pure enantiomers, and we also carry out chiral HPLC analysis, and we correlate the peaks that we see to the bought and to literature values. And for the rest of them, we also simply compared them to literature values. The retention times by chiral HPLC.
You bought racemic mixtures, right?
Yeah.
Not enangiopure. You bought racemic mixtures, right?
Of these, we bought enangiopure and enangiopure mixtures, and not mixtures, sorry, pure compounds. And of these, we only had racemic mixtures. But these we just carried out the reaction, and we we extracted the product and carried out chiral HPLC analysis.
So let me understand. You had pure enantiomer, which you compared with your reaction mixture, right?
Yes.
Hello? Okay, you had pure enantiomer, which you compared with your reaction mixture, right?
Yeah. Well, in this case, we had pure enantiomers, so we could analyze the kinetic variable for both of them individually. And for these, we just carried out this reaction. So if it's selective, we will enrich one enantiomer, and this we can analyze by extracting this product and analyzing it via chiral HPLC. And this will tell us which enantiomer we have.
Okay, then that's okay. And then the second question, of course, you will answer. But now, can you also maybe try to give that enzyme a substrate that you don't know the stoichiometry and see how it will behave?
Of course you can. So I think your first question was, can we also get the other stereoselectivity? And
yes,
the answer is yes. We can challenge our library with the converse enantiomer here, and during the screening process, we will then filter out enzymes that have a different selectivity, hopefully, if they are in the library. And you could put any substrate here that you were interested in. That's what you also did with the substrates that I showed you on the Outlook. We've already seen activity. We already have activity with our variant here, but on selective, and we've seen activity in the droplet sorting system for other substrates as well.
Okay, so just moving on to, we have quite a few questions coming in on you. So when you let me know when we have to stop this. So we have a question from Benjamin Basanta. So you should now be able to talk, Benjamin.
Can you hear me?
Yeah.
Yes.
Okay. Great talk, Aaron. Thank you very much. That was very interesting.
Oh no, he's muted again.
Oh. Okay.
You were muted for a second there.
Okay. Can you hear me now?
Yeah.
Okay. Yeah, I noticed you did take into consideration the one of the native amino acids, proline there. If I understand correctly, because you thought it might have an important structural role. Yeah. So would you care to comment a little bit on, in general, what your approach is to the type of mutations and positions you allow? Do you just choose things around the active site? Do you avoid a certain type of residue or secondary structure? And if you could design your ideal structure to put a binding site in, what would you like it to be?
So I think in this case, what our goal was actually to limit our codon set as much as we can and still have a certain diversity in there. So we have small hydrophobic, large hydrophobic. And this pocket is all inside the enzyme. So all of these residues are very hydrophobic. And so that was the first design thought that we have. And by limiting these codon sets to very, very narrow set of amino acids instead of codons, so it's just 6 codons, the goal of this project was to target a large part of the active site. Actually, here you can see these are almost all residues that make up this side of the active site. And the other consideration is, of course, if you just use Pymol, for example, you can place your substrate such that the hydride that has to be transferred to the FAD needs to somewhat point into direction of the FAD, and you can then figure out where you would expect clashes. Yeah.
I see. Thank you.